So, this is Dr. Stanley Plotkin, and I'm going to speak on the history of vaccine development and the current situation in the field of vaccinology. Obviously, this presents my views and not those of anyone else. So if the listener wants more information about the subject that I will speak about, I recommend these sources. There is a chapter in the vaccines textbook by my wife and myself. There are two short books on the subject of vaccine history, edited by Artenstein or by myself. And finally, there is a big book on the subject by Hervé Bazin, written both in French and in English, which will tell you everything you wanted to know. I'd like to begin with this quotation from the first edition of the vaccines textbook, which summarizes the importance of vaccines to public health. The impact of vaccination on the health of the world's people is hard to exaggerate. With the exception of safe water, no other modality has had such a major effect on mortality reduction and population growth. And that statement is as true today as when it was first written. So, starting with variolation, the use of scabs from smallpox infections to prevent smallpox. This technique started probably in China in the 15th century, and its first use was by nasal insufflation. That is, scabs would be ground up and the powder insufflated up the nose, and this would in general render people impervious to exposure to smallpox. But in the Caucasus, to which it spread, the method used there was largely by scarification. In other words, making a incision in the skin and placing smallpox scab material in the skin. This picture is of Lady Mary Wortley Montagu, the wife of the British ambassador to Turkey, who saw the method being used in that country and who brought it back to England and made propaganda for its use, getting the royal family to use it, and then also watching the technique of variolation spread in England. And this was certainly a good way of preventing serious smallpox. However, there were obvious difficulties in making this method general. But Voltaire, when he was exiled in England, saw variolation being done and wrote back to his French audience an article called the 11th Philosophical Letter on variolation. And in this letter, as illustrated here, he amusingly wrote about the beautiful Caucasian women who were favorites of the harem of the Sultan, and that variolation was used to prevent those women from getting smallpox and spoiling their beauty. But more seriously, Voltaire was definitely in favor of variolation as a method of preventing smallpox. But as we all know, the first real vaccine was developed by Edward Jenner in the UK. And he, as well as a number of other people, noticed that milkmaids maintained their complexions because they seldom got smallpox. And the reason for this, Jenner determined, was because they became infected with cowpox from the cows they were milking, and that this protected them against smallpox. So he began to use cowpox obtained from lesions in milkmaids to vaccinate. And of course, the root of the word vaccine refers to the cow. Here is a diagram from one of his papers showing a lesion on a milkmaid's hand, which he would then use to vaccinate others. The widespread use of the vaccinia virus was certainly able to pretty much wipe out smallpox in Western Europe. The vaccinia strain, the origin of the strain is somewhat mysterious in that it is not really cowpox. It probably is closer to horsepox. At any rate, it took many years before the next step was taken, and that was by Louis Pasteur, who reasoned that if one could in some way weaken pathogens and use them as to inoculate people, that they would prevent the disease that the pathogen would normally cause. He started with bacteria and showed in the laboratory that that was the case in animals, but of course became very famous for attenuating rabies virus. Rabies was then a frequent disease in France. There were many rabid dogs and wolves, and his experiments with both bacteria and the rabies virus established vaccinology as a field of scientific development. This is a picture showing one of his first bacterial vaccines. This was a vaccine against anthrax. Essentially, it was a heat and oxygen-killed anthrax spore vaccine that he used in a demonstration experiment to prevent anthrax in sheep and goats at a place called Pouilly-le-Fort outside of Paris. This was a public experiment, and the result was so striking. All of the control animals died and the vaccinated animals survived, that Pasteur instantly became famous, and the idea of vaccination was clearly demonstrated. But even more fame came to Pasteur because of his efforts in the field of rabies. He was able to show in animals that he could gradually attenuate rabies virus by using exposure to oxygen. And when in 1885 a child was brought from the Alsace who had been bitten by a rabid dog, he decided to try the vaccine on this child in sort of a post-exposure vaccination. Rabies vaccine can be used post-exposure because the virus has to attach to the nerve, has to replicate in the subcutaneous tissue and then attach to the nerve, and that can be prevented by antibody to the virus. So Joseph Meister survived. And also an older boy, Jean-Baptiste Goupil, who had protected children by strangling a rabid dog, also came to the Pasteur and was also vaccinated successfully by Pasteur. This brought great fame to Pasteur and to the Pasteur Institute. Essentially, enabled the foundation of the Pasteur Institute because of the success of rabies. The next great discovery was made in the U.S. when Daniel Salmon and his colleague Theobald Smith inactivated salmonella and showed in animals that such an inactivated preparation could protect them against inoculation with salmonella. The laboratory at the Pasteur Institute simultaneously also made this discovery, leading then to the development of inactivated bacterial vaccines. One of them was a typhoid vaccine by Wilhelm Kohle in Germany, and another vaccine by Sir Almroth Wright in the UK. Now, these vaccines were somewhat reactogenic, and it is said that the British troops going to South Africa for the Boer War at first refused the typhoid vaccine and indeed threw it overboard from the ships that they were taking to South Africa. But eventually Roth showed by more or less case control experiments that the vaccine really worked, and therefore that the typhoid vaccine became widely used and was indeed one of the first killed bacterials. Pierre-Paul Émile Roux, one of Pasteur's co-workers who had been important in the rabies experiments, discovered that the diphtheria bacillus produced in the medium a toxin, that is, something that was toxic for animals. And this important discovery, then, the development of antitoxin by Emil von Behring in Germany, worked his Japanese co-worker Shibasaburo Kitasato. And this, of course, was a demonstration of the importance of antitoxin or antibodies in prevention of disease. It was Paul Ehrlich in Germany who really developed the concept of antibodies, that is, a substance in the blood that you could induce by injecting with various antigens of importance, and that these antibodies could then protect against disease caused by the organism in question. Jules Bordet, a Belgian scientist, was able to show that detection of antibodies was frequently made easier if a complement was put into the reaction, so that the importance of complement to the function of antibodies was shown. And he was also responsible for cultivating pertussis bacilli in artificial media. Here is a timeline showing the development at the end of the 18th century, and also the developments at the end of the 19th century with killed bacterial vaccines against typhoid, cholera, and the plague, all of which were shown to be useful, but which of course have been replaced by better vaccines. Gaston Ramon, in about 1926, realized that he could inactivate tetanus and diphtheria toxin using formalin, and this made it much easier to immunize animals and humans against those bacteria, and eventually leading to the toxoids that we use today to protect against tetanus and diphtheria. Meanwhile, again in the 1920s, Calmette and his colleague Guérin were passaging Mycobacterium bovis, the bovine tuberculosis organism, in artificial medium with the objective of developing a vaccine against tuberculosis. The vaccine was first used by oral administration to babies, but after some years, the use of intradermal administration of the organism became more regular, and this type of vaccine was shown to be highly effective in children to prevent disseminated tuberculosis. So this was an important example of attenuation by passage in artificial medium. Now, pertussis organisms were inactivated by various people in the 1920s, but the development of a really effective vaccine is attributable to these three women, Pearl Kendrick, Grace Eldering, and Lonnie Gordon, working at the Bureau of Biologics in the U.S. They were able to develop methods to cultivate the organism, to passage it in culture, to purify it, to inactivate it, and to show that it was still immunogenic. And that resulted in the pertussis vaccine, which came into use in the 1940s. Meanwhile, in Africa and in South America, yellow fever was causing great epidemics and high mortality. French workers attenuated yellow fever virus in the mouse, but this was still highly reactogenic, and it was Max Tyler, a South African physician, at first working in Africa, but then at the Rockefeller Institute in New York, who was able to attenuate a strain called 17D, which was both highly effective and well tolerated. This won him a Nobel Prize, and the vaccine, which of course is still widely used today. As far as influenza is concerned, the great epidemic in 1918 showed the importance of the disease, but it was hard to cultivate the virus until Richard Shope in the 1920s was able to isolate the virus from swine. Later, workers in the UK and Wilson, for example, as well as Thomas Francis in the US, isolated the human virus in chick embryos, and this eventually led to the original vaccines against influenza. So this timeline shows the 1920s and 1930s, the advent of the toxoids, the BCG vaccine, the first pertussis vaccines, yellow fever, and influenza. an activated influenza vaccine. But perhaps the most important discovery of the 20th century was that of cell culture by these three workers in Boston, John Enders, Frederick Robbins, and Thomas Weller, who showed that they could grow polio and other viruses in cell culture. This led to an explosion of vaccines. Hilary Kaprowski was the first to use cell culture to grow polio strains, which were attenuated in culture and became the first oral polio vaccine. Jonas Salk used polio virus grown in cell culture to develop the inactivated vaccine, which did so much to control polio in the U.S. and other Western countries. And Albert Sabin used cell culture to attenuate the three types of polio virus and to make a trivalent vaccine, which has been widely used and has brought polio to the verge of eradication. Meanwhile, back in Boston, John Enders was cultivating measles virus, and with Samuel Katz attenuated measles virus in cell culture, leading to the Edmundston strain that was then further attenuated and has become the standard measles vaccine strain today. Maurice Hilleman, working at Merck, was one of those who further attenuated measles, but who also isolated mumps virus from his daughter and attenuated it in cell culture and combined it then with rubella vaccine to make MMR. He also isolated hepatitis A virus, made a vaccine, and was one of those made hepatitis B vaccine. Now this timeline shows the 50s through the 80s, showing the development of polio vaccines, measles, mumps, and rubella. Rubella vaccines were developed in the 1960s, and the strain that is used in MMR was developed by yours truly. Anthrax vaccine was made not by inactivating spores, but by taking the secreted toxin, protein toxin of the organism and activating it, and that has been a potent vaccine. As far as hepatitis B is concerned, the first vaccine was derived from human plasma, that's plasma of infected individuals, by Maurice Hilleman. And that was effective, but of course presented certain dangers. I'll come back to that in a moment. But meanwhile, because of antibiotic use, the mortality from pneumococcal pneumonia and pneumococcal invasive disease had gone down. But Robert Austrian, working in Philadelphia, showed that pneumococcal disease was still prevalent, and that if you purified polysaccharide capsules from the bacteria, you could prevent pneumococcal disease. He, working with Hilleman, developed a 23-valent polysaccharide vaccine, which could be used in the elderly to prevent invasive pneumococcal disease. However, that vaccine would not work in young infants who do not respond well to polysaccharides. Or people had to go back to the discovery by Oswald Avery earlier in the 20th century that if polysaccharides of bacteria were conjugated chemically with a protein, that they became more immunogenic, and that they then would be immunogenic in infants. And John Robbins, working at NIH, was the first to develop a protein conjugate vaccine against Haemophilus influenza type B, the cause of much meningitis in children. And that ultimately led to the development of vaccines against all forms of common meningitis, as shown in this timeline. If you look at the 1980s, you can see the Haemophilus influenza type B, and then the conjugate, and this method of conjugation was subsequently used for meningococcal vaccines and for pneumococcal vaccines. But still another important strategy of vaccine development was developed in the 1980s, and that was the result of a discussion between Herbert Boyer, shown here, and Stanley Cohen, allegedly at a delicatessen lunch when they were both attending a meeting in Hawaii. And they talked about the possibility of genetic engineering, that is to say, inserting genes for certain proteins in Escherichia coli and letting the bacteria produce the protein. This method, of course, worked and led in the field of vaccinology first to the hepatitis B vaccine, which is used today. Rather than the plasma-derived vaccine, it was much safer to purify hepatitis B vaccine produced in vitro, hepatitis B surface antigen produced in vitro, to vaccinate people against the disease, which was done by Merck and GSK simultaneously. Going back to live virus vaccines, Michiaki Takahashi in Japan attenuated varicella virus, which has been used in particular in the U.S. and which in effect has controlled varicella in the U.S. Again, in the 1980s, it was shown that a group of viruses called rotaviruses, because of their structure looking like a wheel, were the main cause of infantile gastroenteritis. Several groups set out to develop vaccines. The first success was by Albert Kapickian at NIH, who used the method of reassortment. That was based on the fact that the rotavirus genome is segmented, and therefore if you grow two rotaviruses together, you can make, in effect, hybrids. So what he did was to take a rhesus rotavirus, which he thought was attenuated for humans, and grow it together with human rotaviruses of 4 serotypes to make an attenuated rotavirus vaccine based on reassorting the segments of the RNA genome. Unfortunately, the rhesus rotavirus was not sufficiently attenuated and caused intussusception, or or otherwise called intestinal invagination. This problem was solved, as I will show you in a moment. But here is the timeline for the end of the 20th century, showing For example, the development of inactivated hepatitis A vaccine, acellular pertussis vaccine based on proteins purified from the bacteria, rotavirus vaccines in the rotavirus vaccine, the first one in the end of the 90s, the meningococcal conjugate vaccine I mentioned, and the pneumococcal conjugate vaccine. So by the end of the 20th century, practically all invasive pediatric bacterial infections could be controlled. After 2000, however, in the 21st century, a number of important vaccines have also been developed. A live attenuated influenza vaccine, in other words, the influenza virus, human influenza virus, has been attenuated by passage in cell culture and also reassortment with an attenuated strain, and that is used mainly in children. New rotavirus vaccines, and I'll show you who developed those in a moment. Quadrivalent meningococcal conjugate vaccines, widely used in U.S. adolescents. Human papillomavirus, and I'll have more to say about that. A zoster vaccine, which is essentially the varicella vaccine with a high titer. A cell culture Japanese encephalitis vaccine, and a meningococcal group B vaccine. I'll have more to say about that. The current rotavirus vaccines were developed in two laboratories. One, my own laboratory, through the work of Fred Clark and Paul Offit, using, again, reassortment, but this time using a bovine virus as the attenuated donor, which is indeed highly attenuated for humans. Also, a human rotavirus strain has been attenuated in cell culture by Richard Ward and David Bernstein in Cincinnati. As far as papillomavirus vaccines are concerned, it was realized that these viruses cause many different cancers, both in women and in men, and that the vaccine against those those viruses are extremely important. It was difficult to make a vaccine, but Ian Fraser in Australia demonstrated that the L1 protein was the important protein in a vaccine, or could be an important protein in the vaccine. And Doug Lowy and John Schiller at NIH in the U.S. developed a practical method of making virus-like particles from the L1 protein of papillomaviruses, and their discovery has led to the vaccines that we have today, which are already bringing papillomavirus cancers under control. So I should mention before talking about this slide the meningococcal group B vaccine. This vaccine was developed by Rino Rappuoli in Italy, using essentially genomics, that is to say, by analyzing the genome of meningococcus B and selecting those parts of the genome that code for proteins that protect in mice, that is, vaccinating mice with these proteins, determining which ones are protective, and putting 4 of these proteins together in a vaccine with the outer membrane proteins of a particular meningococcal strain, and that vaccine is now licensed and being used, as well as a meningococcal group B vaccine based on just one of the important proteins of the bacteria. And we are waiting for results of efficacy trials using these vaccines. But this slide I show simply because it graphically illustrates the cumulative number of vaccines developed since the first one I mentioned, the smallpox vaccine. So the pace of vaccine development continues. Now, I want to talk about the vaccine industry first, and the fact that this vaccine is now highly concentrated. There are only 4 manufacturers currently driving innovation: GlaxoSmithKline, Merck, Pfizer, and Sanofi Pasteur. All of those companies are in the U.S. However, there are other important manufacturers who are also doing research and who may contribute to the development of new vaccines. They are listed here: Astellas from Japan, AstraZeneca, CSL from Australia, Johnson & Johnson, which is both in the U.S. and Europe, the Serum Institute of India, now one of the largest vaccine companies in the world, and Takeda, another company from Japan. But I should mention that there are important regional manufacturers in Brazil, in China, in India, in Indonesia, and in Japan. And these manufacturers may ultimately one day contribute to vaccine development and production elsewhere in the world. That being said, I want to emphasize that currently the vaccine development is based on a relatively small number of manufacturers, and although the world of vaccine development research is certainly alive and well, there are many biotechs, but they must link with a producer in order to bring their vaccine to market. The other thing I want to emphasize is the historical development of methods to develop vaccines. There are methods for the development of live vaccines and for inactivated vaccines. For live vaccines, this began, as I mentioned, with Pasteur and his attenuation, physical attenuations, but then moved to passage in animals, eggs, or in vitro, and then in the 20th century, passage in cell culture and various methods in vitro of attenuating viruses or bacteria. As far as inactivated vaccines concerned, well, the first vaccines, as I showed you, were inactivated whole organisms, but then toxoids, inactivated toxins were developed. Then extracts, subunit extracts of parts of bacteria or viruses. For example, the hemagglutinin from influenza or the protective antigen from anthrax. As far as polysaccharides are concerned, I mentioned the use of pneumococcal polysaccharides. The typhoid polysaccharide has also been used as a vaccine, and conjugation of those polysaccharides with proteins, as I mentioned, has resulted in many powerful vaccines. Acellular pertussis vaccine is based on purified proteins from the pertussis organism, and recombinant technology or gene-produced proteins has resulted in the hepatitis B vaccine, the papillomavirus vaccine, and now the meningococcal group B vaccine. But there are even newer methods for attenuating. Reverse genetics, so-called, which was the method used for meningococcal group B, but in addition, temperature-sensitive mutations can be induced in organisms, and viral recombinants and deletion mutants made so as to attenuate viruses. One can use codon deoptimization to cause attenuation. The replication enzyme can be altered so as to produce more faithful replication and therefore more attenuated forms of virus. One can use microRNA insertion to attenuate, and a lot of attenuated vectors, viruses and bacteria are being used to present or to produce proteins from pathogens by inserting the genes for those proteins into the vector. And one can even deliver genes that code for proteins by using invasive but attenuated bacteria. And as far as inactivated vaccines are concerned, DNA plasmids and self-replicating RNA are both being actively investigated as ways to stimulate immune responses to important proteins that are transcribed and translated by the nucleic acids. Reverse vaccinology I've mentioned. Antigen identification by transcriptomics and proteomics will identify antigens that are important for vaccines. Fusion proteins can be made now, fusing important proteins for induction of protective responses. The development of new adjuvants, including cytokines, is a very important field now for improving the immunogenicity of vaccines. And the stimulation of innate immunity is another way of improving immune responses. So all of these discoveries, together with the older methods of vaccine development, are meant to erase this memory. Here is a stone from a cemetery in New England that I found, and it shows a stone inscribed in the memory of 4 sons who all died early in life. This kind of tragedy, which Jabez and Lydia Sweet underwent, is fortunately much less likely because of the development of the vaccines that we have talked about. Thank you.